Hello and welcome everybody to my channel Omini Bio Entrance. Today we will be discussing the next 2017 question paper. I will be discussing only the biology section. So this section contains 15 questions. For questions 2.1 to 2.10, only one of the four options is correct. The correct answer will earn 3 marks, a wrong answer will earn -1 mark, and an unattempted question will earn 0 marks. So coming to the first question. A person is infected with a deadly microorganism. Choose the option that would provide the fastest immune response against the microbe in the affected immunization. Option A, active immunization. Option B, passive immunization. Option C, immunization with recombinant DNA. Option D, antibiotic administration. Now what is active immunization? It is the process by which immune system of the body is stimulated when exposed to a particular antigen. Body produces antibodies against the antigen. Then what is passive immunization? Passive immunization is direct administration of antibody to fight against the antigen. Then what immunization with the recombinant DNA involves administration of a particular DNA segment which produces antigen. Then what is antibiotic administration? Antibiotic administration is done in order to fight against microorganism. Now the person is infected with a deadly microorganism and passive immunization will be the fastest, because in this antibodies are directly administered which will fight against the microorganism. So option B is correct, passive immunization. Consider two unlinked genes P and Q. A plant with genotype PPQQ was crossed with another plant with genotype p to obtain F1 progeny. If the F1 progeny is test cross, the percentage of resultant plants with the genotype PPQQ will be. So answer is 25%, because after the test cross you will get the ratio 1:1:1. So out of the 4, since this is a dihybrid cross, out of the 4 progeny you will get 1 recessive. So this is the cross, these are the parents, that is capital P, small q, capital Q, crossed with small p, small q. The gametes are PQ and PQ, and F1 progeny is capital P, small p, capital Q, small q. Now this is test cross, the F1 progeny is test cross. Test cross is nothing but crossing with the recessive parent, and you will get progeny in the ratio 1:1:1:1. So they have asked for the percentage of the resultant plants with the genotype p, small q. Out of these 4 progeny, you have only 1. So 1/4, that is 25%. So answer is option B, 25%. Now next one. The mean biomass per unit area and the net primary productivity of 4 ecosystems P, Q, R and S are tabulated below. Okay, the 4 ecosystem most likely would be tropical rainforest, temperate, desert and algal bed. So these are the various 4 combination of options they have given, that is tropical forest, desert, temperate, grassland are involved in all the A, B, C, D options in different combinations. So we have to see for the correct combination. So the primary the net primary productivity will be highest for the tropical rainforest. So here there are two ecosystems, that is Q and R, where the net primary productivity is 2000. But here the mean biomass per unit area of R is more. So naturally that must be tropical rainforest and this must be the algal bed. So R is tropical rainforest. Yes, option C. R is tropical rainforest, as well as option D, and Q is algal bed. So option C can be ruled out because Q is temperate grassland, but actually it will be algal bed. So option D will be more correct. And the lowest net primary productivity will be for the desert ecosystem, that is 71. So S is 71 and R will be, no sorry, P will be temperate grassland. So answer is option D. The next one, each male gamete of a coconut has 16 coconut plant has 16 chromosomes. The number of chromosomes in a cell of the white pulp of coconut is. So the gamete, that is the male gamete of the coconut, has 16 chromosomes and male gametes are haploid. So N is equal to 16. Then they have asked for the number of chromosomes in a cell of the white pulp of the coconut. The white pulp of the coconut is nothing but the endosperm of the coconut, which is triploid. So it will be 3N. So the number of chromosomes in a cell of the white pulp of coconut is 3 into 16, that is 48. So answer is option C, 48. Next one. The relative percentage of nitrogenous base of the genetic material isolated from various species P, Q, R, S, T are listed in the following table. Choose the correct statements from the following. So they have asked for single-stranded DNA, RNA. Okay, I can see double-stranded RNA also, single-stranded RNA, double-stranded DNA, single-stranded DNA. Okay, now we have to choose the correct answer. Now coming to species P, you can see that adenine matches with thymine and guanine forms hydrogen bond with cytosine. So here in species P, adenine and thymine number are equal. Similarly, guanine and cytosine are percentages are equal. There is no uracil, so it is DNA and it will be double stranded. So P is double stranded DNA. Q also the same way, adenine percentage equal to thymine percentage, guanine percentage equal to cytosine percentage. There is no uracil, so naturally this is a DNA and it is also double stranded. So P and Q are double stranded DNA. Now R, coming to R, the percentage of adenine doesn't match with that of thymine, and similarly for guanine and cytosine also variation is there, but there are no uracils, so R must be single stranded DNA. So if we see here A, B, C, yes, as per A it is PQR contains single stranded DNA, no. PQR contain double stranded RNA, no. PQR cannot contain single stranded DNA, C is not right. So option D will be more correct. That is species P and Q contain double stranded DNA and species R contain single stranded DNA. You will see for species S. Species S, the adenine is equal to uracil, guanine equal to cytosine, so and there is no thymine, so it is an RNA and it is double stranded. And T will be adenine not equal to uracil, similarly guanine not equal to cytosine, so it will be single stranded RNA. So option D is correct. Then, in order to estimate the gross primary productivity of a pond containing phytoplankton and zooplankton, a student collected water samples from the pond in two labeled bottles X and Y. The dissolved oxygen of the samples immediately after collection was 8 mg per ml. The dissolved content was measured after 24 hours of incubation was as follows. Bottle X 2 mg per ml after incubation in dark, and bottle Y 18 mg per ml after incubation in light. So there is variation in the dissolved oxygen, what has been measured after 24-hour incubation in the dark. Now they have asked to estimate the gross primary productivity. So gross primary productivity is net photosynthetic activity plus respiratory activity. Now what is respiratory activity and photosynthetic activity, how we will come to know. So when the bottles X and Y containing pond water were kept in the dark and light, there was a decline in the dissolved oxygen in bottle X. Why? Because phytoplanktons are not able to photosynthesize in the dark condition and oxygen will be used up in respiration. So bottle X is having less dissolved oxygen than when incubated under dark conditions. And for bottle Y, there was increase in the dissolved oxygen because photosynthesis is taking place in the presence of light in the production of oxygen. So the dissolved oxygen of bottle Y is more after incubation for 24 hours in the in the light. Now from this data we can calculate respiratory activity, that is initial dissolved oxygen minus dissolved oxygen after incubation in the dark. You will get the respiratory activity. So 8 minus 2 will be 6 mg per ml. And net photosynthetic activity will be dissolved oxygen after light incubation minus initial dissolved oxygen. So 18 minus 8 will be 10 mg per ml, and Gp will be sum of net photosynthetic activity and respiratory activity, that is 16. So answer is option C, 16. Now next one. Consider the following statement regarding Michaelis-Menten constant. Km is indicative of the affinity of an enzyme for a substrate. Now that is correct. Now a high Km indicates high affinity for substrate binding. No, a high Km indicates low affinity, while low Km indicates a high affinity for substrate binding. Then Km is a measure of the substrate concentration required for effective catalysis to occur. So first statement is correct, second is wrong. Now we are coming to the third one. Third one, Km values will measure as the substrate differs because different enzymes have different affinity towards different substrates. So statement 3 is correct. So answer is option 1 and 3 are correct, and that is C. A piece of liver tissue is homogenized in a laboratory, and the homogenate is subjected to sequential centrifugation at increasing speeds. Centrifugation is used to separate subcellular components based on size and density. The steps carried out in the experiment are shown below. So homogenized liver tissue centrifuged at 500 G, you get sediment 1 and supernatant 1. Then again you centrifuge it at higher speed, that is 10,000 G. You get sediment 2 and supernatant 2, and again at 25,000 G you get sediment 3 and supernatant 3. Now sediments 1, 2 and 3 would most likely contain. So the greater the density, the faster it moves. Okay. So when homogenized liver tissue is centrifuged at 500 G, the heaviest subcellular component will settle fast. So it will be nucleus, which is as per from these options, that is nucleus, mitochondria, ribosome and lysosome. Nucleus is more denser and it will settle fast. Then comes at 10,000 G comes mitochondria, that will also settle next to nucleus, then ribosomes and lysosomes. And the third one will be naturally lysosomes, because there is no other option with the first one as nuclei and second one as mitochondria. So third one will be lysosomes. So option B is correct. Then next one. The presence or absence of certain subcellular components in the cells of 3 organisms are given below. Ribosomes, microtubules, mitochondria, lysosomes, vacuoles. P, Q and R are most likely to be. Now if you see from P, ribosomes are present, microtubules, mitochondria, lysosomes all are present. So naturally it is eukaryotic. And many small sized vacuoles are present, so it points out to animal cell. So from these options A and B we can see, C and D can be ruled out. The next one, ribosomes are present but all the other cellular organelles are absent including vacuoles. So it must be prokaryotic, so Q must be E. coli. So option A is correct. We will see for the other one also. Ribosomes are present, other organelles are also present, and single large sized vacuole is present. So naturally it is a plant cell. So option is garlic, R. So option which is correct is A. P frog, Q E. coli, R garlic. The next one. The concentration of dissolved carbon dioxide in blood varies in different blood vessels. Choose the most appropriate option with decreasing order of dissolved carbon dioxide concentration in the blood vessel. So the first one has to be the one with highest concentration of carbon dioxide, and that is naturally the pulmonary artery. It emerges from the heart and carries blood with low oxygen content and high carbon dioxide content into the lungs. So pulmonary artery will be the first one. So B and D can be ruled out, and we will see for A and C. In A and C we have skin capillaries, that is pulmonary artery greater than skin capillaries, then pulmonary artery greater than aorta. Okay, skin capillaries will have more carbon dioxide than aorta. Why? Because there is some amount of respiration. Carbon dioxide levels are increased and are a bit higher than that in aorta. So the next one will be skin capillaries greater than aorta, then alveolar capillaries. So option A is correct. Then questions 211 to 215, one or more than one of the four options may be correct. Your answer is regarded correct only if you choose all the correct options and no incorrect options. A correct answer will earn 4 marks and a wrong answer or an unattempted question will earn 0 marks. So the question 211. Two pedigrees P and Q are given below. Individual 1 in F0 generation of the pedigree Q is not a carrier of the trait. Circles in the pedigree represent females and squares represent males. Filled shapes indicate affected individuals while unfilled shapes indicate unaffected individuals. So if you see pedigree P, we can see that unaffected parents have got an affected child. So it must be somewhere related to a heterozygous condition for parents. That is, parents genotype will be heterozygous. That is, suppose if capital A is for dominant trait and small a for the recessive trait, and if the recessive trait is causing the disease or the dominant trait is causing the disease, then the if the dominant trait was causing the disease, then heterozygous parents would also have been affected with the disease. But from the pedigree analysis in the F1 generation, we can see that only one individual out of the 3 are affected with the disease, and the parents are heterozygous. So it must be naturally a autosomal recessive trait. Okay, so P must be autosomal recessive trait. Then we can see that it is not present in all the generations. Then the trait is occurring in male as well as in female. There is nothing like only females are having the disease or males are having the disease, females are carriers of the disease, nothing like that. It is just like any male or female can come in contact with the disease. So the pedigree P shows an autosomal recessive trait. Now coming to pedigree Q. Pedigree Q, unaffected parent is not transmitting the trait to the offspring. So that is because here the F1 F0 condition, the male is affected by the disease, and to whomsoever the male has given the gene of it has been expressed in males and female children. That is, half of them have got infected with the disease and the other half have not been infected with the disease. So if you consider this one, that is the female to be having small a genotype and the male to be having capital A small a, then you will see that half of the individuals will be having capital A small a trait and the other half will be having small a small a. So half of the children will be affected and the other half will be unaffected. So it is a dominant trait. then the trait is occurring in male and female with almost equal frequency. So it is not an X-linked inheritance. So pedigree Q will be autosomal dominant trait. Now we will see the options. So both pedigrees P and Q represent autosomal recessive trait. Now only P is having the autosomal recessive trait. Then both pedigrees P and Q represent autosomal dominant traits. No, P and Q both don't represent autosomal dominant trait. Only Q represent autosomal dominant trait. Then pedigree P represents an autosomal recessive trait and Q represent an autosomal dominant trait. Now that is correct. Then next one, pedigree P represents an X-linked recessive trait and Q represents an autosomal recessive trait. So that is wrong. So option C is the only one which is correct. Then next one, the matrix for the presence or absence of certain features in vertebrates P, Q, R, S and T are given below. Presence of a character is denoted by 1 while absence is denoted by 0. The statements that hold true is or are. Q could be either a rat or a gorilla. So what is Q? Q is having 4 limbs, 4-chambered heart, amniotic egg, hair, fur, yes. Q can be a rat or a gorilla, so that is correct. That is mammalian characters are given for Q. Then P most likely represent a bird. Then what is P? P 4 limbs, 4-chambered heart, no. Amniotic egg, no. Hint, hair or fur, no. So P is not at all a bird. Bird is having an amniotic egg also. So P is wrong. Then T could be a lizard. So we will see what is T. It has 4 limbs, 4-chambered heart, no. Amniotic egg, yeah, it's a reptile. Hair, fur, no. So T is also not correct. Then next one, S could be snake, vertebrate, 4 limbs, 4 limbs 0, 4-chambered heart 0, amniotic egg is there and hair or fur is absent. So S could be a snake. So answer is option A and D. A student wants to construct a plasmid backlight that when transformed into a bacterium will produce light if and only if the sugar lactose is present in the medium. For this, she uses the following DNA sequences: promoter of repressor, promoter of LAC-Z, operator and repressor gene LAC-I from the lactose operon, and a structural gene X which encodes a protein that produces light. The different DNA elements 2 to 5 and vector 1 were digested with restriction enzymes and ligated to produce the plasmid. The DNAs used along with the restriction enzyme sites at their ends are given below. Slash in the sequence represent the point at which restriction enzyme cuts the DNA. Choose the correct sequence of DNAs that can possibly be ligated to produce the plasmid backlight. Now a student wants to construct a plasmid backlight that will encode the protein that produces light, and she has chosen the elements of lac operon or the lactose operon. She has taken the repressor, promoter of LAC-Z, operator, repressor gene LAC-I of the lactose operon, but she has not chosen the structural genes of the lactose operon. In place of the structural gene, she wants to have her own structural gene that which encodes the protein that produces light, especially in the presence of sugar lactose. So they have given the DNA elements and their restriction enzyme sites they have given, and we have to choose the correct sequence of DNA. Now one thing you have to remember is that when you are choosing for ligating the vector, the 5' end should get ligated to the 3' end. So that will be the order of ligation. So the first one is the vector sequence, that is at 5' end you have GGATCC and at 3' end you have TCTAGA. Now TCTAGA occurs at the 5' end of 2 DNA elements, that is second and third. But so from this we have to choose the next which will be ligated to the vector. So after the vector, if you take PI LAC-I, then the other ending sequence will be GGATCC. That will get ligated to vector backbone at the 5' end, and the plasmid will be completed. So your plasmid will have only the vector backbone and pi laci. There won't be other genes like PZO and gene X. So your vector will be incomplete. You should incorporate these genes also. So second option can be ruled out. Now coming to third option. So after the vector, if you ligate TCTAGAPZ, then you will get a much more sensible plasmid. So GCTAGC, you have it over in your DNA structural element 4, that is 5' GCAGC, and the 3' end of this operator sequence is ACTAGT, that is matching with DNA element 5, that is gene X. The gene X has got at its 3' end TCTAGA, that matches with the second DNA element, that is pi laci, and the ending sequence is GGATCC, that can be ligated to the vector sequence itself because it is having GGATCC at its 5' end. So the sequence, the correct sequence of DNAs that can be possibly ligated to produce the plasmid backlight will be vector PO, gene X, pi laci, and the vector itself. So option B is correct. Then ecologist David Tillman grew laboratory populations of two species of diatoms, namely Asterionella formosa and Synedra ulna, in liquid media supplemented with silica. The researcher monitored not only the population growth but also the level of silica in the medium. The result of the experiment are shown in the graphs P, Q and R below. P and Q respectively are graphs obtained when Asterionella formosa and Synedra ulna are grown separately. Q graph is obtained when both are grown together. The correct conclusion that can be drawn from the result is RR. Now before coming to that, if you see the graph P, okay, if you see the graph P, you can see that graph P is of Asterionella formosa, that it maintains a steady state of growth. See here, it maintains a steady state of growth, that too at silica concentration of 30 micromoles per liter. And graph Q shows that this also maintains a steady state of growth but at a lower silica concentration, that is 20 micromoles per liter. But when both are grown together, you can see that Synedra ulna is reducing the silica concentration to 20 micromoles per liter and thereby inhibiting the growth of Asterionella formosa. Okay, so this one, that is Synedra ulna, is an inhibitor of Asterionella formosa. So the conclusions we can read. When grown together, presence of AF in the medium has positive effect on the growth of SU. No, the AF doesn't have any positive effect. It is having a negative effect from SU. When grown together, SU reduces silica concentration to a level that does not allow AF to multiply. That is correct. B is correct. Then AF reduces resource availability at a faster rate than SU. No, AF doesn't reduces any resource availability. So C is wrong. There is a competition for the resource between AF and SU. That can be seen from the graph R. So D is also correct. So options B and D are correct. Then next one. The number of double-stranded DNA molecules of an autosome varies at different stages of the meiotic cycle. Choose the correct option. So option B is correct. Why? Because meiosis is a reduction division. The double-stranded DNA are reduced to half at the end of meiosis 1 and meiosis 2 is equational division. And The number of double-stranded DNA will always be half in meiosis 2 as compared to meiosis 1. So at the interphase stage, you can see number of double-stranded DNA 2, metaphase 1 also it will be 2. But when it comes to metaphase 2, it will be 1 and telophase 2, it will be 1. So option B is correct. Thank you. Please like, share and subscribe my channel. Thank you for watching.